MRI of immunotherapies. So I just want to start. This is not quite up to date, but a relatively recent picture from my lab. We have a lot of undergraduate students in particular, as well as grads and postdocs in a research tech. So just a little bit, because I'm sure we're a small center and not necessarily many people know about us. So in Halifax, we have a 3T MRI with a PET insert that allows us to do simultaneous PET MRI. We have a small SPECT system. We also have another standalone PET system that was designed for CL2 and CL3 PET imaging. So we can use that either in the imaging lab, or it can actually be taken into the level 3 lab for use with high level pathogens. We also have an older trifoil PET CT, although we really don't use the PET on it anymore. We just use the CT. So just a little bit, kind of an introduction for people who aren't familiar. Immunotherapies are a way of getting your body's own immune system to attack cancer. Cancer is very good at co-opting your immune system to either shut it down, to protect it sometimes, or to basically pretend it's not there. So there are a number of different types of therapies that have been developed, kind of far too many to talk about in a talk like this. They're basically kind of aimed at 3 different areas. One would be to enhance your body's T cells. So some kind of therapy like adoptive T cell therapies, where they take your body's T cells and inject them back in after they modify them, that will help drive those T cells to attack cancer. There is blockade of immune inhibition, because your body is really good at shutting it down via things like Tregs and MDSCs. You can actually block these cells from working by depleting them or by inhibiting them. You can also do what's called enhanced tumor antigen recognition. So tumors, not always, but often have very specific cell markers that are unique to the tumor, or at least pretty different from other normal cells in the body. So if you can get cells, the T cells, to recognize this, they tend to be much better at recognizing and killing tumors. So a lot of people wonder why we can't just study immunotherapies the way we study therapies like radiation therapy, surgery, chemotherapy. But the problem is that immunotherapies work in a much more complex fashion, and they don't behave the same way. In particular, normally when you apply a therapy like chemotherapy or radiation therapy, you're going to get tumor shrinkage after you do treatment. With immunotherapy, that's not always the case right away. You can actually get what's called pseudoprogression, where you get tumor swelling because of an influx of a lot of immune cells. So the question that doctors ask when they see this is this real progression of the tumor, or is this pseudoprogression? And it's a very important difference. So there are a number of classic criteria for kind of assessing if you're responding to cancer therapy or not. The RECIST criteria is one of them, where it's basically has different classifications depending on tumor volumes, but they don't work for immunotherapies. So there's a lot of different groups developing their own. But regardless, it's pretty clear that it's not as clear cut as it is for chemotherapies and radiation therapies. They're pretty complex. On top of that, immunotherapies are highly individual. A therapy that cures one person may not work at all in the next person. And we really don't understand why, even now. We don't understand how to predict responses, and we don't understand how to combine therapies. So right now, if you want to combine different immunotherapies, the overwhelming way to do it is to guess. So generally you take a therapy that worked or kind of worked and you add another therapy that you think kind of works with it. In a lot of cases, we do not understand the mechanism of action. So how can we use imaging to study these cancer immunotherapies? In our lab, there are about 3 different ways we study it. Obviously, there's a lot of other different ways I'm not going to get into. So the 3, I would say, most common ways are, 1, to develop novel imaging probes. So with both MRI, PET, and things like this, these are all the people that are looking at really interesting and new probes that can help assess molecular effects. I'm not going to discuss that one in this lab. We don't do a lot of that, although we are planning to do more in the future. I'm going to talk mostly about number 2 and 3. So number 2 is actually directly imaging the therapy and the effects that this therapy has on local anatomy. And I don't just mean looking at changes in volume. So we want to look at the behavior of the therapy. We want to learn more about the mechanism of action. And we really, in a lot of cases, want to develop imaging biomarkers. It would be really nice in a lot of cases if we can have prognostic biomarkers as well as diagnostic. But prognostic is extremely helpful. We don't know who's going to respond best to immunotherapies. If there is some kind of imaging-based biomarker that can help, that can save a lot of money and a lot of time for the patient. And then the third one is imaging immune cells. And I would say this is probably the biggest area of research in our lab, and looking at migration and interaction with cancer and the therapy itself. So the first project I'm going to talk about is actually about using antigen tracking. So this is work we've done over a number of years with a local pharma company called IMV. So IMV has their own proprietary immunotherapy. So they inject what is called DPX. And this formulation is actually, instead of kind of spreading systemically through the body, it's usually injected subcutaneously and will actually remain at that location for a significant period of time. What happens is antigen presenting cells are drawn to the site, will take up the drug, will actually migrate to lymph nodes and activate and program T and B cells. These T and B cells will then circulate, infiltrate and recognize the tumor and release antibodies and peptides, or in the case of T cells, directly attack cancer cells. And it can be personalized by using different antigens for different types of cancer. Other types of cancer vaccines that have been developed are, some are very simple, like a water and oil emulsion, where you take an antigen and some kind of adjuvant that will help enhance the immune response, and then you suspend it in oil. That is another way of kind of taking it and making it localized and holding it for a long period of time to present these antigens to the immune system, because you don't want it to disperse quickly. So one of the things that we did to start is we took this antigen and we labeled it with iron oxide. We wanted to use MRI because obviously it's non-invasive, but most importantly, it would let us look at both individual responses and longitudinal studies. Imaging has that really nice advantage of being able to track things over time. So we used SPIO, specifically a 30 nanometer nanoparticle that was able to just attach to peptides via pretty simple click chemistry. The downside to SPIO is that it is a negative contrast agent, so you get signal loss. But it is still very visible in the regions we're interested in. As you can see, on the left image is our control. On the right is when we have iron oxide labeled cells in this particular instance. We also did immune assays as a tumor experiment just to make sure that there was no difference in immunogenicity or tumor response when we added iron to this antigen. So first thing we did is we added it to the oil for one group of mice and to the antigen for the other. So in the case of the oil, we actually tracked it for 6 months. And we found that it remained at the site of injection for at least 6 months. And we had to actually stop at 6 months because the mice were getting too big to fit in our coil anymore. For the antigen, you see a stronger drop in signal in the first 15 to 21 days, and then a slower sustained response over time. We repeated it in tumor-bearing mice and saw very similar things. We obviously can't go out as long with tumor-bearing mice. But the most clearance happens in the first 3 weeks, but you actually can detect clearance for at least 6 months after injection, which is really important for maintaining a sustained immune response. It's also interesting to note that we were able to track the migration of this iron oxide labeled antigen to the lymph nodes, which is really crucial because one of the things that we assume is that antigen presenting cells are showing up at that site of injection and taking that antigen to the lymph node. And in our cases, it was, and not only that, it was doing it over a sustained period of time. And we know that it's not just, it gets there at day 12 and then it sits there. This iron oxide is small enough that it will get digested generally within 7 to 10 days. So for example, the iron oxide that is there at day 26 is not the same iron oxide that was there at day 12. So we got continuous clearance, which is, again, really important to understand. We noticed this for at least 4 weeks. And we saw it in at least 80% of the mice as late as 4 weeks post-injection. So then the next thing we wanted to do is we wanted to understand, how do other platforms work? So how does this water and oil antigen combination work? So we did a tumor challenge as well as looking at the iron clearance. And so in this particular study, the DPX worked better. The mice that were treated with DPX had smaller tumors. But most interestingly, it had a slower clearance than the water and oil emulsion, significantly over basically the entire study period. So the water and oil emulsion, the antigen in that, although it does last longer than, say, just injecting with PBS, it is cleared much more quickly than in the DPX, which is likely part of the reason for that stronger immune response. So again, there are lots of other types of platforms, these are not the only ones. So we looked at a couple of other ones. One is called squalene oil and water. Another one's a saponin liposome. One of these is similar to how the flu shot is actually presented. In most cases, we found DPX actually had smaller tumor volumes, significantly so. There wasn't as strong a difference in terms of antigen clearance. We found that kind of overall, they had similar types, although there was a significant difference between DPX and the squalene. Interestingly enough, and I'll come back to this in another slide, is that the saponin liposome mixture actually caused a really strong response in the immune system very quickly after injection. So if we vaccinate or inject at day 0, by day 7, we were seeing a lot of swelling in the area. The lymph nodes just grow hugely. And so we saw over 1,000% increase in the volume of that lymph node. But then it tapers off very quickly. Whereas DPX kind of actually has a slow increase in those lymph node volumes on the draining lymph node. To kind of show you what it can look like in terms of the inflammation, that saponin liposome vaccine, as indicated in the red areas, induced a lot of overinflammation. So the black is our labeled antigen. That bright white signal is actually inflammation in that area. You also see that inflammation happening in the lymph nodes as well. The problem with too much inflammation is that it acts as a T cell sink. So instead of getting the T cells into the antigen and having them pick up, actually see antigen from these antigen presenting cells, they kind of get trapped there and they don't go to the tumor, which is where we want them. So there is kind of a fine line to walk. So it's important to understand how these therapies work. So we can learn a lot about this kind of drug behavior by using imaging. We saw better infiltration of antigen presenting cells when we looked at the slides, the histology from these ones, and we saw this sustained antigen presentation. We also saw really significant differences in lymph node swelling in response to treatment, which is really interesting because something like that could be a prognostic biomarker, maybe not immediately, but still earlier than waiting for tumors to shrink. Right now, we're kind of repeating this with transgenic mice models because we want to understand what immune cells are actually involved in this kind of picking this antigen up and taking it to the lymph nodes. And, by eliminating certain populations of cells, will this actually affect the rate of our clearance? All right, so now I'm going to move into cell tracking. And so the last two projects that I discussed are related to cell tracking. So the first one is ovarian cancer. So in immunotherapies, there's a lot of different types of immune cells that are really important. And the more cells that you study, the better off you are. There are two classes, basically, generally speaking. There's the immune activating cells like cytotoxic CD8 T cells, dendritic cells, or antigen presenting cells. And then there are the immune suppressing cells like regulatory T cells and myeloid derived suppressor cells. Unfortunately, actually, most of the work in the literature on tracking immune cells is just on immune activating. So I want to emphasize that if anyone is thinking about this, I actually encourage you to study a lot more of the immune suppressive ones. And I'm guilty of it too. So just, ovarian cancer is leading, it's one of the worst types of cancer for women in terms of death. They can start in either the ovaries or the fallopian tubes. And there are 3 different kinds of cancer. Epithelial tumors are the most common. Unfortunately, they're the most malignant generally as well. And so the model that we're using is an epithelial cell tumor. Part of the reason we're using this model is actually because, again, we did this in partnership with DPX and or IMV, who makes DPX, and they're actually testing it in clinical trials. And so one of the things we wanted to do is replicate some of the early clinical trials to see if we get similar results, and specifically see if we can explain some of the anecdotal findings that they're seeing. So this is a combination therapy study. We looked at DPX Survivac, which I mentioned before. We also looked at low-dose cyclophosphamide, which is basically just low-dose chemotherapy. And we also used what's called a checkpoint inhibitor, or anti-PD-1. We wanted to look at tumor growth and immune cell recruitment. The reason we chose this combination is to match that clinical trial. So we wanted to try and quantify immune cells in the tumors. So specifically, we used a sequence called TurboSBI. So TurboSBI can be used to indirectly obtain the number of cells in vivo. So again, we're using SBIO, which induces the signal loss due to the iron, specifically in R2 star change. You can theoretically use R2 star to quantify concentrations of SPIO. Now, there are a lot of caveats that I'm not going to get into today. There are a lot of assumptions you have to make, which are not always valid. But as an overall kind of estimate, it works reasonably well. So it's a reasonably quick sequence developed by collaborators of ours, James Ryu, and as well as students in the lab, obviously. 3D quantification of cells in about 25 minutes. Now, it's important to note that it doesn't work with free iron. The iron has to be taken up by cells because it uses that difference in the ratio of R2 star and R2 that happens specifically in iron labeled cells. So you can see that what happens is you get a signal intensity. You can measure the R2 star from the slope. You need a calibration curve, and this has to be done in vitro so that you know approximately what concentration of cells responds to what R2 star. And you also have to know what a background is as well. So for example, on the left is the anatomical, and on the right is the anatomical plus the map of cells, where the cells are kind of in bright green. And you'll notice they're actually pretty clustered on the edge. This is actually very common in tumors where you don't get penetration into the center, because in the center of these tumors, a lot of times they're dying. And so you get cells kind of ringing the periphery of the tumor. So in this study, we had donor mice that were treatment matched. So we would take the cells from the donor mice, label them with iron oxide up, and then inject them into the recipient mice. And like I mentioned, all the mice were treated with those 3 therapies. So, just to start, we did see a significant difference. There wasn't a significant difference at the time point, but when doing an ANOVA, there was a significant difference over time. And there was an interaction between treatment and time, which is interesting. They aren't completely separate variables. At day 56, or from day 42 to 56, we saw a significantly greater increase in the volumes in untreated tumors. So kind of the longer you wait, the bigger the difference you see between them. You can see on the right is actually a PET scan we also did, where the tumor is highlighted in the white dots. The one on the left is treated, the one on the right is untreated. So for the cell tracking itself, we looked at 2 types of cells. One was cytotoxic T lymphocytes, or CTLs. The other was myeloid cells. So for the CTLs, we actually saw a significant decrease in treatment, and particularly at later time points. So in untreated mice, we saw far fewer cytotoxic T lymphocytes present in the tumor, even though the tumors themselves were much larger. And so the density decreased significantly. We looked at the lymph nodes. I'm not sure why this is auto scrolling. We looked at the lymph nodes. There were some trends, but nothing was significantly different. Then we looked at myeloid suppressor cells, saw very similar results. Untreated cells, or untreated tumors, had far fewer of these myeloid cells as compared to treated cells. Whereas in lymph nodes, we didn't see any significant difference. We actually looked at the volume of that tumor draining and vaccine draining lymph node. And for treated mice specifically, we did see that the DPX draining lymph nodes were bigger. And so the ratio of the 2 lymph nodes was actually significantly larger than in untreated mice, which again indicates that it might be, even as early as day 42, kind of a prognostic biomarker. So we looked at this combination therapy, which tells us a lot, but the reality is we want to kind of take a step back in the future and look at the therapies individually. We know it slows primary tumor growth. We know it increases the recruitment of these 2 immune cells to the tumors, which is really helpful. And it increases swelling in the DPX draining, that inguinal lymph node, which again could act as a potential biomarker for therapeutic efficacy. So the last thing I'm going to talk about is kind of work we're doing now and work that's kind of going into the future. And it actually leads a bit into one of the student talks Anna will be giving just after this. So we are really interested in moving from doing a single cell at a time to looking at multiple contrast agents. Like I mentioned earlier, the more contrast agents you can do, or basically the more types of immune cells you can study or mechanisms you can study, the better off you are. These immunotherapies have a lot going on. They're very dynamically complex. On top of that, again, ideally qualitative is useful, quantitative is more so, because it lets us understand kind of what is happening over time. So the one on the right, this image is what we got from our turbo SBI. So again, the one that we use for our ovarian cancer and other early models. We'd like to add a GAD contrast agent. Gadolinium is obviously very commonly used clinically. You can use it to study a lot of processes, you know, non-targeted like perfusion or dynamic contrast enhancement. There are a lot of specific probes that are targeted, things like targets for caspase in apoptosis or targeted to proteins. And we can combine that with SBIO, which you can use for tracking a number of different cell types. Currently, generally quantitative MRI is restricted to a single contrast agent. Not necessarily SBIO, it's also fluorine-19 is very commonly used, but still a single agent. Although there are some groups starting to work with fluorine and SBIO, which is really useful, I think. So what we're proposing is actually using magnetic resonance fingerprinting to do gadolinium and iron at the same time. And so I'm not going to describe this in detail because this is actually what Anna's presentation is about after mine. But just in general, MR fingerprinting, or MRF, allows us to quantify two different contrast agents that are administered essentially at the same time. You need two different parameters that you're measuring. In our case, we need T2 star as well as T1. Previous work has been done separating gadolinium and manganese, just using T1 and T2, as you can see this figure on the right. However, dual quantification of T2 star contrast, I should say, hadn't been done before. So Anna published a paper on that in 2021 as our first attempt at doing this. So our goal, our final goal, is to quantify both T1 and T2 star simultaneously, ideally in 3D, but particularly a large volume. We want it to be quite high resolution, and we want it to happen in about 20-25 minutes, which is pretty reasonable for my scanning. So what we're interested in studying in this particular model is glioblastoma. Here's kind of a sample image on the right from an MR. Glioblastoma is extremely aggressive. It's the most common primary brain tumor. It grows so quickly. And on top of that, it grows with brain tissue. So it kind of intertwines with normal tissue and vasculature. You can almost, I kind of describe it sometimes as students as kind of a graspy octopus, in that it reaches into all the surrounding tissues, and it's really hard to distinguish between the two at the edges. Right now, treatment options are surgery, radiation, and chemotherapy. That being said, even if you get treated, your average survival is 12 to 18 months. Only 25% of people with this live more than a year. Only 5% of people are going to live more than 5 years. So glioblastoma is essentially a death sentence. And it really comes down to how quickly and what is your quality of life going to be. Because again, anything in the brain is going to cause significant cognitive and motor issues. So we need better treatment options. Immunotherapies are one of those. So gadolinium is very interesting in the brain because many gadolinium contrast agents will not penetrate through the blood-brain barrier unless there is damage. So in this particular case, we used multi-HANCE, which is used clinically. It will not enter the BBB or will not pass through the BBB if it's intact. However, glioblastoma clearly damages the blood-brain barrier, which will allow gadolinium to enter. And so you should theoretically get increasing amounts of gadolinium infiltration correlating with increasing amounts of blood-brain barrier damage from the glioblastoma. For iron oxide, in the past, we'd always done in vitro labeling and then injecting of the cells. In this particular case, we want to inject the iron oxide intravenously where it will spread throughout the body. It is small enough to enter the brain. It will get taken up by macrophages, both within the brain and in other parts of the body. And macrophages in other parts of the body can be recruited to the brain as well. If we quantify iron oxide, we can quantify macrophages in the brain throughout the growth period. The SPIO we use is quite small, and that 30 nanometers, and it's generally broken down in 7 to 10 days. Theoretically, if you wanted to, you could use a larger one, but then have to contend with blood-brain barrier issues. So again, there's kind of a trade-off. So in our group, to do this, you have to do brain surgery. So you have to implant these brain cancer cells. In our case, we used a glioma 261 model directly into the brain. They mimic the natural disease progress quite well, actually. The microenvironment looks very similar. You get kind of that natural growth. It just happens on a much quicker time scale than in humans. So you immobilize the mice in a stereotactic device, drill a small hole, inject 2 microliters of cells, and then monitor the mice closely. You do have to monitor this model really well because the tumors grow rapidly. Mice can be totally fine one day and then kind of exhibiting various motor defects the next. So you can't leave this model very long. So we looked at 3 different groups. One, our control group, another with that checkpoint inhibitor, anti-PD-1. So one of the only clinically approved immunotherapies, which is why it's being studied so much. And we also use what's called temozolomide. So temozolomide is the clinically approved chemotherapy for glioblastoma. It damages DNA, but more importantly, has immunomodulatory effects. So we use lower dose TMZ to try to help boost the anti-PD-1. We looked at twice weekly images, pre and post the iron injection for the immune cells, and because MRF does require a pre and a post. We also digested brains at the end of the study to investigate immune cells. So for the MRI, we did MRF, and we did just a standard T2 weighted FSE. We did this theoretically up to 65 days. Realistically, you're not going to get longer than day 42. So there was a significant difference in survival with treatment. Interestingly, there was a significant sex difference with treatment. So anti-PD-1 and TMZ treated females, which is the black line on the left, they actually had the highest survival rate of all of the groups, followed closely by anti-PD-1 only females. Whereas regardless of treatment in males, they all died at a much quicker rate. And you can see just kind of some of the tumor volumes and how quickly they grow in these mice. There are some mice that need to be terminated as early as, day 15, day 16 in this model. So here's kind of an example of some of the images from this. So the top left is the T2 weighted FSE. The blue circle indicates the area with a likely brain tumor. On the top right, you see a FDG PET image we get at the same time, indicating that there definitely is brain tumor in that area. And then this is a particular map of macrophages, generally through the brain, as well as gadolinium. So we looked to quantify gadolinium infiltration into tumors, and not surprisingly, the larger the tumors, the higher the mass of gadolinium. And this was, a very strong linear effect, and quite significant as well. And this was true for pretty much all treatment groups, as well as we didn't really see a sex difference with this one. Slopes were a little bit different, but no significant differences. Macrophages were a little bit more complex. The effect was not necessarily linear for some of the groups. For some of them it was, some of them it wasn't. So this is obviously kind of a more complex behavior that we're still investigating. Overall, you do generally see an increase in iron oxide with tumor volume, but there are a lot more outliers. So overall, the behavior was a lot more variable, which is interesting. Here's an example of a couple of mice. Specifically, these are female. The one on the left is untreated and the tumor is in green. And so you see kind of some tumor-associated macrophages in this particular mouse. And on the right, you actually see a lot of macrophages, a lot more macrophages, higher density, and just throughout the whole brain in this particular mouse. However, we saw a huge amount of variability even within groups. So one of the biggest problems that we have with analyzing all this data is just in how much data we have. We do longitudinal imaging, so that means generally at least weekly. We are getting multiple types of contrast agents. So we have contrast agent maps as well as just T1, T2, and T2 star maps, and an FSC. We also are looking at different treatments. And so the amount of data we're working with balloons pretty quickly, even with small numbers of mice that are used in preclinical studies. And so the question is, how do we analyze this data the best way? And so one of the things that we're working towards is using radiomics. Radiomics is used a lot clinically, but it's really underused preclinically right now. So for those who aren't aware, just for radiomics, biomedical images have a lot of information that reflects underlying pathophysiology. And the specific pathophysiology often depends on the contrast. So you can convert these images into mineable high-dimensional data. You can correlate them with clinical outcomes to increase precision. You can potentially develop prognostic or diagnostic biomarkers, prediction of therapy response. There's a lot of options. So some of the features that you can look at are really simple, like, what is the volume of the region of interest? In our case, the region of interest is probably, say, the tumor or the brain. What is the sphericity? What is the surface area? Generally, kind of what are the region of interest shape features? Then, of course, there's the first order features. Things like the mean are what we were using before. It's a very simple feature to get. But there are a lot of other first order features that might be helpful as well. Maximum, minimum, median, standard deviation, variance, those are all very useful. And then there's texture features. So this has to do with spatial arrangement of the pixels. And this is very interesting in tumor imaging. Tumors are extremely heterogeneous. And knowing how the spatial arrangement changes over time can give you a lot of insight into dynamic behaviors of the tumors as well as the immunotherapies. So this is a sample pipeline, and Laura, who's the other student presenting, is actually going to talk about this in more detail. But generally, you get all your maps, you have to segment them, and then you start looking at features and the algorithms that are necessary to kind of identify your classifiers and just identify biomarkers. And how do you assess this? What is your accuracy? What is your sensitivity, specificity? So where are we going in the future with this? Because I've kind of touched very briefly on each of these. I think in particular, molecular imaging is really underutilized in the drug development pathway. So there are so many types of imaging. Obviously, we're all MR people. But we can just learn so much, and they have such advantages for individual elements of drug effects and interactions. And being able to monitor longitudinal effects is just so crucial. And we can study, not only the effects of the drug on the body, but also how the drug itself interacts with the environment, whether it be the tumor or the body as a whole. Another area that we're kind of pushing into in the future are things like respiratory viruses. So obviously, COVID made a huge impact on society. But the flu, RSV, and coronaviruses in general are extremely understudied with imaging. That being said, they have their own challenges. For some reason, SARS-CoV-2 is actually still a level 3 pathogen, even though you're more likely to get it from the grocery store than you are from actually working with it in the lab. So we're hoping it eventually comes down to a level 2 before too long. But that means that you have to have the equipment in a specialized lab, and there are very few places. London is one of them, but there are very few places that have type of imaging equipment. But we need better tools. Everyone thought at the beginning of the COVID pandemic that COVID was a respiratory only virus, but now we're looking at neurological effects, long COVID, brain fog. What happens to people who have this? We don't understand a lot about this. And if we want to develop safe and effective vaccines, and if we want to, design better therapies for people, we need to understand these complex interactions. So I always kind of end my talks encouraging people to get into the virus areas. So I think that's pretty much it for me. I just want to thank everyone in my lab, as well as funding from places like NSERC and our local Beatrice Hunter Cancer Research Institute.